Hello everyone, and welcome to episode 121. In today's show, we're starting a new series on the crustaceans, and we're going to begin with the story of crustacean evolution. This should be a supremely interesting episode, and frankly, every episode in this series is going to be mind-boggling and wicked cool. Every group of crustaceans that we're going to look at has something unique to offer. Something weird or incredible or just downright fascinating. The crustaceans in general are an awesome group of animals. They're extremely diverse, with a wide variety of forms, more unique and varied than even the arachnids. They're ecologically important. They play a vital role in the animal food web, especially in the oceans. And I mean what I'm about to say next in the most affectionate and endearing way possible. But they're fucking disgusting. They smell bad, they look like creepy little aliens, their facial topology is characterized by lots of fleshy flaps and protruding tentacles, they have way too many legs, their slimy exoskeletal skin folds stiffly and weirdly at their joints, and they've evolved all manner of spines, spikes, barbs, claws, and pincers, from the small to the very large. But how did they get here? From what dark, watery corner of the oceans did they arise? And what events, what factors, what evolutionary forces separated the ancient crustaceans from their ancestral kin, the myriapods and the arachnids, and shaped them into what they are today? These are the questions we'll be addressing in this episode, as we explore the complex and incredible story of the evolution of crustaceans. Now before we get started, let me clarify some things and explain some of the context of our research and our knowledge of crustacean evolution. Much like the primitive arachnids and the myriapods, the interrelationships within the crustacean clade, their internal phylogeny, has not been conclusively determined, and there's lots of debate about what group is related to what other group and to what degree. Before the development of modern scientific instruments and genetic analysis, species were compared and taxonomically organized morphologically, based on their physical appearance and structure, and sometimes, to a lesser degree, on their observable behaviors and life cycle. When genetic sequencing and other kinds of molecular analysis were developed, new phylogenies were constructed based on this more accurate molecular data, based on mitochondrial DNA, on ribosomal RNA, or gene families and alleles, and so on. Usually, these molecular studies clarified mysterious relationships and corrected the earlier phylogenies constructed with relatively crude morphological analysis. This new frontier of molecular research gave us a more accurate and more comprehensive picture of the evolution of many different lineages of all kinds of organisms, animals, plants, fungi. And for the most part, this advancement was extremely edifying and has led to enormous developments in our understanding of evolution and the interrelationships of all life. However, in a minority of cases, these molecular studies haven't provided much help at all clearing up relationships between clades. And in the most frustrating cases, the molecular studies have actually made things more complicated and less clear. This is the case for many of the arthropods. As we discussed in previous episodes, the relationships between many basal arachnids remains shrouded in mystery. We don't know precisely how the Apillionese harvestmen, the Xiphosaura, the Acariforms, the Solifugi, the Palpigradi, and so on are all related. This is to say, we're just not exactly sure who came first, or in what order their ancestors emerged and diverged, and who diverged from who shared a common ancestor with who, and so on and so forth. The myriapods are similar, although the challenge here is a little less substantial simply due to the fact that they have much lower order and family level diversity. Very, very recent molecular studies have suggested a bi-pronged family tree, starting with the ancestral myriapod stem group, which then split into one branch that produced the pauropods and the symphylans, and the other branch split into millipedes and centipedes. This conclusion, based on molecular research, conflicts with myriapod phylogeny hypotheses that had existed for several decades before, and which were based on cruder morphological studies and very early genetic analyses. And the crustaceans are perhaps the worst offender. When most of the clades had been sampled and sequenced, and the first crustacean phylogenies were put together, things were so much worse. It was really confusing how all of the morphological relationships that we thought existed were totally unsupported by the genetic data. To make matters worse, trying to parse out some of the relationships in the genetic data itself, it was really difficult and hard to decipher. Over 2 decades of research, the molecular phylogeny has been reformatted over and over again, sometimes with major changes. In that sense, the early evolution of the crustaceans is somewhat of a mystery. It wasn't until the crustaceans evolved harder exoskeletons, and when the larger malacostracans evolved in the late Cambrian, around 500 million years ago, that they began to leave behind a lot of fossils. Like, a lot of fossils. The myriapods, in comparison, have a super weak fossil record, with almost no fossils left behind. There are some orders that are represented by a single-digit number of fossil specimens. The myriapod fossil record is just abysmally poor. In contrast, the crustaceans have an extremely extensive fossil record, starting from about the Carboniferous period, with fewer fossils remaining from the Cambrian and Ordovician. This is perhaps due to their hard carapace, and their tendency to live in silty marine areas where they can be easily preserved in mud or sediment. As a result, the evolution of later crustaceans and the phylogenies of malacostracans, which make up about 4/5 of all modern crustaceans, these can be reconstructed with robust confidence. It's just the early evolution of the crustaceans that's still poorly understood. It's still shrouded in mystery. And with that said, this is where our story of crustacean evolution begins. In this mysterious era at the dawn of the Cambrian, sometime around 570 to 550 million years ago, before even the true myriapods had come into existence. It's believed that the common ancestor of myriapods and crustaceans existed around this time, near the latter half of the Ediacaran period. Rising from some late Proterozoic lineage of multicellular creatures, it would have been elongated, with a segmented body and many pairs of legs, with a head equipped with 2 pairs of antennae, or some kind of foremouth appendages, as well as a pair of maxillae, or miniature arm-like appendages near the mouth that helped to handle and process food. This Ediacaran common ancestor would have scurried along the seafloor, probably feeding on detritus and marine snow, although it may have been a carnivore. It's a bit difficult to say, as millipedes are generally herbivores, and centipedes are generally carnivores, but many primitive crustaceans, for example, like the Ostracoda, include herbivores, parasites, detrivores, generalists, and carnivores. It's likely that this ancient common ancestor of myriapods and crustaceans was an omnivore, a kind of generalist scavenger that grazed on aquatic fungi and mats of bacteria, while occasionally catching a smaller creature like a nematode, a protozoan, or a lophotrochozoan, which was a type of small proto-mollusk that may have been evolving around this time, 570 to 550 or 540 million years ago. This period of time was the later half of the Ediacaran, the geological period immediately preceding the Cambrian. It was a 93 million year-long stretch of time that saw some of the earliest multicellular life diversify into primitive lineages like sponges and cnidarians, in addition to less familiar organisms that characterized the Ediacaran biota. Many of these strange and very, very primitive creatures are unlike any kind of organism alive today, and they're believed to be a collection of stem group lineages who did not survive through the late Ediacaran or through the early Cambrian. The gradual extinction of almost all of the Ediacaran biota at the end of the period opened up many ecological niches, allowing the smaller, less dominant species to rise to the fore and replace the older, extinct Ediacaran species as the dominant grazers and predators and whatnot. This is probably about when, in the late Ediacaran, during this great ecological turnover, the common ancestor of myriapods and crustaceans emerged. It would have been one of the rare Ediacaran groups to survive the end-period extinction and proliferate in the Cambrian. And by the time the Cambrian explosion came to an end, about 15 to 25 million years after it began, all the major phyla that we know and love today were in existence. 510 to 520 million years ago, this common ancestor had fully diverged into the ancestral myriapods and the ancestral crustaceans. And before the Cambrian had come to an end, pretty much all of the major crustacean lineages had been established. However, they were not all equally represented. In the context of crustacean biodiversity, the Cambrian was dominated by members of the primitive Oligostracca. Although more complex and more derived lineages like the Malacostraca existed in the later Cambrian, they were not numerous or widespread, and they wouldn't become numerous or widespread, or particularly biodiverse for that matter, until much, much later. By studying the anatomy and behavior of the myriapods and the modern crustaceans, we can gather clues as to how they may have diverged. The myriapods leaned into their linear segmented body plan, and they adapted to the terrestrial habitats on dry land. The ancestral crustaceans were once thought to have had a similar body plan, being roughly myriapod-like. But later genetic analyses has thoroughly reinforced the basal nature of the Oligostracca. And this is important because the Oligostracca are stubbier and more oval-shaped. They're not long chains of body segments like the myriapods are. This has led to a re-evaluation of the myriapod crustacean common ancestor. Now, the current thinking is that the common ancestor was short-bodied, shaped kind of like a grain of rice, and all of its descendant lineages, myriapods and crustaceans alike, evolved variations in the number and structure of the body segments, so they weren't long to begin with. The many body segments in a linear body plan, that evolved later. To put this another way, the common ancestor of crustaceans and myriapods was short-bodied, and after it diverged, the new myriapod lineage rapidly evolved a more elongated and heavily segmented body form, which served them well when they colonized dry land and began to inhabit the new soil habitat. The crustaceans, in contrast, developed more evolutionary variety, and they produced lineages with a wider range of body segments. Now, generally speaking, they've slowly evolved to have more body segments. That's the general trend. Most lineages are more likely to add a body segment over time, rather than eliminate one. That's not to say it doesn't happen. Some lineages certainly have lost body segments, but the general trend is increasing numbers of body segments. But as you can tell by all of my hedging here, there's lots and lots of variety within the crustacean body plan. In fact, when we include the hexapods, as the hexapods are descended from the crustaceans, we're talking about the most biodiverse clade on the planet, and the evolution of the number, size, shape, and arrangement of their body segments is a defining and fundamental part of their evolution, both collectively and as individual classes, orders, and families. Another interesting fact is that the crustaceans evolved biramus limbs. Yeah, there's a cool new vocab word for you. So the millipedes, in contrast, have uniramus legs, una meaning one. The leg appendage is a series of segments attached end to end in a single continuous strand and a single continuous appendage. The crustacean limb is biramus, and this means that it has a small modification, but this has had enormous evolutionary consequences. The modification was a single branching point somewhere near the base of the limb. Instead of the limb being formed of a single strand of segments, in the crustaceans, after a couple segments from the body, the limb actually branches, and two strands of segments grow outwards instead of just one. This biramus setup allowed each strand of segments on each limb to adapt its own specialization or function. Let me illustrate it this way. If humans had biramus limbs, kind of like crustaceans, then you could imagine we might have a second forearm coming out of each elbow, and that second forearm might have, let's say, fewer fingers, but the fingers are longer so that they're more dexterous and we use them for like fine motor activity. Or maybe it's the other way. Maybe we have the same number of fingers, but on that other forearm coming out of each elbow, the fingers are much larger and stronger. The forearm is more muscular, and it's intended for moving heavy items or self-defense. It's built up and overdeveloped for that reason. That's what biramus limbs might look like on a human, and hopefully that gives you some sense of what this might look like on crustaceans. Now, in addition to all of this, the crustaceans also evolved 2 sheets of carapace on their backs to cover up and protect the clusters of legs on either side of the body. At the front of the body, the tentacles and antenna and mouth parts protrude outward into the environment, and at the rear, the last few body segments make up a tail that pokes out a short distance. We can clearly see this body form in some of the oldest crustacean or stem group crustacean fossils ever known, like the Canidaspis and Perspicaris. These organisms existed in the Middle Cambrian around 505 million years ago. Perspicaris was probably an active swimmer, suggested by its longer tail and the posterior spikes or fins. The known species were between 1 and 2.5 millimeters long. They were quite tiny little things swimming around in these Cambrian oceans. The other fossil species, Canidaspis, generally was a bit larger, at about 10 to 15 millimeters long. They dwelled on the seafloor, sifting through the mud looking for clumps or colonies of microbes to sweep up with their spine-covered appendages. Protective spines existed on its head and around its eyes, which were mounted on short stalks. Their biramus limbs showed significant specialization. The first branch on each limb was like a simple leg, but the second branch on each limb had flattened and broadened out like a fin. In effect, they were just bristling with appendages, with legs for crawling and more fin-like parts for moving mud or for swimming. There's an old evolutionary offshoot, a lineage that emerged in the mysterious darkness at the dawn of the Cambrian, and which left no fossils until the Ordovician, by which point they were already quite well developed and diverse. This ancient branch of the crustacean clade is known as the Oligostraca. In the Cambrian period, it would diverge and begin to diversify on its own, ultimately creating an endless menagerie of descendant clades. The oldest Oligostracan clade, and thus one of the oldest surviving crustacean lineages ever, is arguably the humble Ostracoda. They're also known as the seed shrimp, because, from the side, their bodies have the shape of a small, oval seed. From the front, you can see that they're rather narrow, so they actually have a body shape that's kind of like a small disc. They're not particularly strong swimmers. They get pushed around by the currents, and so they're part of the community of zooplankton, which is a valuable food source for all sorts of larger animals. In the Ostracoda, the 2 sheets of carapace have evolved to almost completely enclose the soft tissues, making the ostracods a kind of rough crustacean analogy to the molluscan bivalves. Protruding from their face and sticking out from between the 2 plates of the carapace are 2 pairs of antennae, which are waved around and used for propulsion to swim through the water, or at least to orient themselves certain ways and to move within the larger current pushing them around. Now, the Ostracoda are really weird because they're kind of like giant gonads with a face. The males, for example, have 2 penises, the result of 3 or 5 pairs of legs fusing together and evolving a sperm-secreting function. The gonads are disproportionately large. In males, these produce similarly large sperm, which is described as coiled up inside the testes. In fact, when the sperm tail is uncoiled, the sperm is longer than the body of the ostracod itself, up to 6 times as long. This is the largest sperm-to-body size ratio in the entire animal kingdom. Ostracod mating behaviors are crude and unsophisticated. The males, which may live or temporarily organize themselves into groups or schools, are swarmed by fertile females. In most species, mating is direct, and fertilization is internal, with the male using one of his two penises. Although a number of species are only females, and they reproduce through parthenogenesis. There's a lot of reproductive variety among the ostracods. For example, some species release their eggs into the water and then anchor them somewhere or hide them, and then they just leave. They leave the eggs on their own. Some species don't even have the decency to bury or hide or otherwise protect their eggs. They just lay them out in the water column, and then if a fish comes by and happens to eat it, well, so it goes. And then you have all of the brooding species, which demonstrate some degree of parental care of the eggs and/or the offspring. In the fossil record, the ostracoda are extremely abundant across pretty much all periods, from the Cambrian to the modern day. They're so abundant that they're actually a kind of biometric for other fossils in geologic strata. Because they're so abundant and so diverse, with so many different forms adapted to so many often highly specific habitats, we can use groups of ostracods, groups with different forms or groups of different species, to measure biomes or habitat zones, especially in shallow water and coastal environments. This general process of using a fossil like this to help date other things is called biozonation or biostratigraphy, where the fossils give reliable clues as to the local environment and the climate of when and where they lived. In this way, the ostracods have been invaluable to developing our understanding of ancient life, not just crustaceans, but all ancient life, as these ostracods are like a nearly ubiquitous kind of biomonitor, which tells us about the world in which they lived, from the general climate and ecosystem at large to the conditions in their very specific habitat. For example, herbivorous ostracods are abundant in shallow water areas with marine vegetation. They live in shallow water areas where sunlight can reach the seafloor, and all of these aquatic plants can grow. These ostracods hang out in these marine forests and these marine fields and feed on the seaweed and the algae and everything else. And then you also have scavenger and carnivore ostracods, which tend to be abundant in the benthic zone, where there's more detritus and microfauna dwelling on the seafloor. There was a really cool 2017 study that analyzed ostracod biostratigraphy in a column of carbonate rock that formed in northeast Brazil. The carbonate rock represented a 25 million year-long span of evolutionary history, from about 40 to 15 million years ago. So, from a geological perspective, these are relatively young fossils. But the scientists identified 119 species of ostracod, most of which were in the Parasocytheridia genus. These species are adapted to brackish water, with an intermediate salinity level between ocean water and fresh water. They lived in lagoons, which were, in turn, surrounded by expanses of tidal flats and stands of mangrove forests. The presence of certain more saline-sensitive species tells us that this was mostly a period of ecological stability, with continual deposition of sediment in the lagoon habitats. At the end of this period, a river had eroded its banks and found a new path to the ocean, which cut right through this fossil bed and turned it into an estuary zone with a higher proportion of fresh water. This killed off or pushed out many of the more saline-tolerant ostracod species. And we can see that in this fossil record, where in the more recent layers of this carbonate rock, we see more freshwater ostracods. Throughout evolutionary history, throughout the last 500 million years, the ostracoda have served as an abundant and invaluable food source near the base of the food web. They would have been eating plant and animal detritus, algae, fungi, and small microfauna like sporozoa and foraminifera. And in turn, they would have been consumed by larger invertebrates like pycnogonid sea spiders and xiphosurin horseshoe crabs. And these in turn would have fed the early vertebrates, like a primitive jawless fish that emerged in the later Cambrian. One of the other major divergences within oligostracans produced the group Mystacocarida. Now, we're not really sure if Mystacocarida is an old branch of crustacean that emerged shortly after Ostracoda, or if this is indeed a subgroup within Ostracoda. That part isn't clear. We're not quite sure what the context of its origination is. But what we do know is that the Mystacocarida subclass is much smaller and less diverse than the Ostracoda, with a mere 13 species across 2 genera, both of them in a single family called Darocylocarididae. One of these genera, Darochylocaris, exists across the Atlantic coastline of North America, from the Canadian Shield down across the east coast to the Gulf of Mexico. The other genera, Tenochylocaris, exists closer to Antarctica, with populations along the southwestern coasts of Australia and the coasts along the temperate southern cape of South America. The Mystacocarida live in interstitial spaces in intertidal habitats. This is to say that they live in between rocks and in crevices and in gaps in the sand and in tide pools in the intertidal zone, the area from which the water recedes during low tide and fills up during high tide. During the recession of the tide, so when it's high tide but the water is going out and it's becoming low tide, many small pools of water are created in clusters of rocks and whatnot. And these are often very rich and exciting microhabitats, with each intertidal pool being home to carpets of algae and other marine vegetation, as well as charismatic sea urchins, sea slugs, clams, barnacles, and even various small fish. Some of these larger animals feed on the Mystacocarid egg sacs, which are deposited into the sand and abandoned by the mother. Mating behavior in the Mystacocarids, up to copulation and post-coital activity, hasn't been observed and scientifically documented, so we can only guess at how that works. The tiny Mystacocarids live in the lower levels of the tide pools and the ocean water, near the sandy sea floor. Much like how the myriad symphylans move between particles in the soil, the Mystacocarids move between grains of sand. And this is because they're really small, typically smaller than a millimeter from the first to last body segment. In shallow water areas, like intertidal zones, sunlight facilitates high ecological productivity, so you have a relatively high density of plants and animals in the coastal communities. This means a lot of living biomass, both flora and fauna, and that means a lot of biomass is eventually going to die and decay and become detritus, or dead biomass that's breaking down into smaller particles, dissolving into the water column where it feeds smaller creatures. Now, the Mystacocarida utilize a really neat filter feeding strategy. Their heads are often heavily equipped with these bristly appendages. The mandibles, maxilla, and antenna, and the carapace of the head segment itself are overdeveloped, and they bear these spines and hairs and bristles. These protrusions are like the teeth of a comb. They can capture this dissolved detritus and extract it from the water or from the seafloor sand. These facial appendages then deliver the food particles to the mouth, where they are ingested. In addition to this really cool filter feeding strategy that's produced a wild and colorful morphology of spines and bristles and hairs, they also use their mouth parts to scrape at algae on the sand or on rocks, and chew into bacterial mats or bacterial colonies. Unfortunately, beyond this, they're a very cryptic group of animals. Almost nothing is known about them, although this is almost certainly due not to their purported rarity or obscurity, but rather to the fact that there's just been relatively little effort invested to research them, compared to some of the more charismatic crustaceans that may be more ecologically or economically valuable. For the Mystacocarids, there are precious few cataloged samples, and even fewer researchers out there in the field actually doing the sampling and collecting and analysis. Because we've done very little research on these guys, we don't really know that much about them beyond technical morphological descriptions and the other big picture stuff that I've reported to you here. But with all that being said, let me speculate a little bit. If the Mystacocaridae appeared after the Ostracoda, and that seems likely based on their low biodiversity and limited geographic range, then I think they may have evolved by unfurling their abdominal segment and developing deeper segmentation in the abdomen, and they reduced or lost the two carapace plates armoring their sides. This freed up space for the newly exposed forelimbs to grow into a forest of hairs and bristles for capturing food. So, in effect, they look kind of like somewhat hairy aquatic centipedes, right down to the rear-facing pair of appendages on the terminal body segment. Now, a third basal lineage, closely related to the first two lineages near the base of the crustacean family tree, are the Branchiura, or the fish lice. There's scant fossil evidence of these creatures until the early Carboniferous, but again, like the Ostracoda, genetic evidence suggests a much earlier origin than what the fossil record indicates. The lack of earlier fossils may be due to their softer shells. They just hadn't evolved shells with a high enough concentration of minerals like calcium and magnesium to readily fossilize, and so when they died, they just dissolved. They didn't fossilize. Anyway, the Branchiurans are roughly similar to their Ostracoda cousins. The Branchiura are also disc or coin-shaped, perhaps a bit more like little ovals with tiny faces, and they have virtually no segmentation on their abdomen. However, where the Ostracoda are shaped like a disc with the flat surfaces on either side, the Branchiura are shaped like a disc with the flat surfaces on the top and bottom. They're also much more dependent on their legs for swimming, as opposed to their antennae, and they show much more pronounced sexual dimorphism. Evolutionarily speaking, the Branchiura grew tired of seeking or hunting for food in the wild and adapted to a more passive, parasitic lifestyle. They saw the emerging metazoan ecology with its worms and mollusks and primitive fish, and instead of looking at these larger animals as potential predators to be avoided, they approached them as novel platforms for shelter and sustenance. This adaptation to a parasitic lifestyle was total, as all known species of Branchiura are obligate parasites that require a host to complete their life cycle. The Branchiurans invade the siphon tubes and the mantle cavities of the mollusks. They invade the eyes and the gills and throats of fish, and when the amphibians emerged and struggled to colonize drier, land-based habitats, the Branchiurans evolved to infest and parasitize them too. Now, the Branchiurans can be divided into 4 genera. One of them, Dolops, is known to use hooks on some of its mouthpart appendages to anchor itself to its host, while the other 3 genera, Argulus, Conopeltis, and Dipteropeltis, have evolved mouthparts and antennae equipped with suction cups to stick to the host. However they do it, once they've securely attached themselves, these fish lice will then start eating some sort of substance or secretion of the host animal. For example, some Branchiurans will feast on the slime coating secreted onto a fish's scales, and they'll also eat the fish scales themselves, or the mucus of a salamander or a sea slug, or the eyeball juices of a large cephalopod. If the skin is generally soft, but also includes crusty patches of dead skin or dead tissue, kind of like cuticles, the Branchiuran will eat those too. And of course, many species of Branchiuran will chew directly into the soft flesh of their host and tap right into the hemolymph or the bloodstream to slurp up nutrient-rich fluid right from the source. Now, as the Branchiurans are obligate parasites that live inside their host, they also end up finding a mate or copulating in or on their host. They reproduce through the use of spermatophores, and once fertilization has been achieved, they part ways. The male disengages and disappears, and the female leaves the host animal to find a suitable place to lay her eggs. Now, this could be virtually anywhere, like on a rock, a coral reef, the shell of a mollusk, or on the stem or leaves of marine plants or mangrove plants. The goal here, the idea, is to get the eggs close to other soft-bodied animals, so that when the larvae hatch, they can easily find some new host to latch onto and parasitize, growing larger and molting until they reach sexual maturity and have offspring of their own. After the female branchiurid deposits the eggs somewhere, usually after arranging them in neat little rows, she'll return to the host animal to look for more sexually receptive males, who are also seeking out new sexual partners on this gory train ride. And the reproductive cycle begins again. There's also a weird mystery clade that I want to mention. Several genomic studies from the early 2010s identified another major oligostrachan lineage, another subclass, sister to the branchiurans. This sister lineage is the pentastomida, or the tongue worms. Like the branchiurans, the tongue worms are larger obligate parasites, usually between 1 and 14 centimeters or so. The pentastomida earned their nickname because of their creepy tongue-like shape. They have an elongated, streamlined, worm-like body covered in a segmented, chitinous cuticle-like carapace. They're like the crustacean attempt at an earthworm, but instead of burrowing into soil, they burrow into flesh and consume blood or mucus. The tongue worms mostly live in the upper respiratory tract, so like the trachea and the mouth-nose region of more derived vertebrates like reptiles, birds, and mammals. They have 4 stubby little appendages with claws on the end, which they use to latch onto the soft tissues on or inside the throat. And they have a tube-like mouth that sucks up the various fluids produced by the host. Here, in this moist, mucousy habitat inside a vertebrate's respiratory tract, the pentastomid finds mates and lays their eggs in the host's soft, sensitive oral or respiratory tissue. The eggs irritate the host, which causes it to sneeze or cough or puke to rid itself of the discomfort. The end result is a cluster of pentastomid eggs suspended in a wad of mucus, splattered on the ground somewhere. The eggs are then incidentally ingested by some other animal, some second host, when it comes to eat up the nutritious mucus or vomit. Think like a fish, a scavenging reptile, or a rodent, or even another oligostrachan crustacean, like an ostracod seed shrimp. The eggs will travel through the digestive system of this second host until they reach the intestines. Here, the eggs will break open and release the larvae, which are surprisingly globular and stubby, reminiscent of the ostracoda and the branchiurans. The larva will then burrow into the soft, pillowy intestinal flesh and create an enclosed chamber or cyst. Within the cyst, the pentastomid will mature and molt occasionally and bide its time. It's waiting for this secondary host to be eaten by a larger carnivore, like a reptile, a bird, or a mammal, which is the primary host that the maturing pentastomid is trying to get to. As it's packaged up in a prey animal's guts like some kind of wild arthropod turducken, when the secondary host is consumed, the pentastomid will burst out of its cyst, almost like it's hatching again from the egg, and invade the digestive tract of the primary host. It will climb up the intestinal tract, up the esophagus, to the trachea, where it will then crawl into the respiratory tract. Here, the pentastomid will have access to mucus produced by the respiratory tissues, and blood from the rich capillary beds. It can also find a mate, some other pentastomid that's also parasitized this unfortunate host. All in all, it's an utterly disgusting parasitic life cycle. Now, earlier, I mentioned that there were some studies from the early 2010s that found that the pentastomida are parasitic crustaceans sister to the branchiura. Curiously, this is not a consistent result, and so this sister lineage idea is actually one of 2 main competing hypotheses for the origin of the tongue worms. The other hypothesis is the idea that the pentastomids are actually much more distantly related, so distant in fact that they're not actually crustaceans. They're not even true arthropods. Instead, they're proto-arthropods, descendants of some ancient stem group lineage that emerged before the first true arthropods ever did, which would have been sometime at least in the late Ediacaran, but probably even earlier than that. This second hypothesis would make the pentastomids more closely related to nematodes and priapulids and other protostome worms than to the actual crustaceans and arthropods. There's several lines of evidence that have driven these diverging hypotheses on the origin of the pentastomids. The body being segmented with a chitin-rich carapace that's replaced through molting all suggest that the tongueworms are at least ecdysozoans. They have an extensive fossil record beginning in the late Cambrian, which is early enough to realistically support both hypotheses. They have similar sperm physiology to the branchiurans, but their larvae have some traits that aren't seen in any other crustaceans or even any other arthropods. To make matters more complicated, genetic and molecular analyses have historically given conflicting results, with some suggesting a more crustacean, sister to the branchiurans origin, while others suggest a more distant origin, perhaps outside of the true arthropods entirely. You'll remember that at the beginning of the episode, I mentioned that molecular studies of crustacean phylogenetics have been perplexing and confounding, often giving contradictory or inconsistent results. There's many examples of this headache-inducing problem, including what I've described right here, the origin of the pentastomids, and their relationship to other crustaceans and other arthropods. So these four groups of oligostracans make up the oldest and most basal branch of the pan-crustacean clade. And the oldest and most basal branch of oligostracan is the tiny seed-shaped creatures of the ostracoda, which are part of the zooplankton, and thus an important part of the food web. Closely related to them are the equally diminutive Mystacocarida, which live in the intertidal zone, crawling or swimming near the seafloor, where they filter feed with their hairy, bristly head. A larger branch of oligostracan are the branchiura, the fish lice. These are a bit larger than the ostracods and Mystacocarids, and they parasitize mollusks, fish, and some amphibians, hooking into them with their mouthparts and consuming their oily secretions, their loose and flaking scales, or their blood. And possibly closely related to the branchiura, although it's not clear yet based on the molecular data, are the pentastomids, the tongueworms, those creepy, slimy, highly segmented parasites that inhabit the respiratory tract of reptiles, birds, and mammals. As the oldest branch of the pan-crustacean clade, all of these oligostracans are sister to the Altocrustacea lineage, which is an explosively diverse clade that contains all other crustaceans. Within the Altocrustacea, there were 5 major divergences, and we're going to cover all of them and the resulting creatures. The first divergence, which occurred sometime in the early Middle Cambrian, created the multicrustacea. These multicrustaceans would become incredibly successful, adapting to a wide variety of habitats and evolving many different forms that are each robustly suited to their habitat. They were so successful, in fact, that about 4/5 of all crustaceans belong to this multicrustacean clade. All the lobsters, all the crabs, and almost all the shrimp have emerged from within the ranks of this ancient multicrustacea superclass. As a result, we'll be spending the next 3 episodes in this series exploring the various branches of the multicrustacea, with an emphasis on the malacostracans, as well as some important bits about their evolution, like the Cretaceous crab revolution and the process of carcinization. We'll get to all of that in the next few episodes. Then, the 5th episode in this series will be all about the remaining major divergences. So after the emergence of the multicrustacea lineage in the early to mid-Cambrian, there was the Allotriocarida lineage that emerged. The Allotriocaridans would produce the horseshoe shrimps of the Cephalocarida in the mid-Cambrian, the freshwater fairy shrimp of Brachiopoda shortly after that, and in the final third of the Cambrian period came the blind and millipede-like Remipedia. The remaining Allotriocarida stock, the sister lineage created by the diverging Remipedia, would go on to produce the Hexapoda, the hexapods. The hexapods would become famous in their own right, for producing organisms as diverse and ecologically valuable as the Protura, the Diplura, and the Insecta. But we'll get to all of these animals in a future series. For now, this was our introduction to the early evolution of the crustaceans and their basal clades. If you enjoyed this episode, then please hit the like button or give the podcast a 5-star review. Hit the subscribe button so that you can listen to new Biologic Podcast episodes right when I post them. If you'd like to support the show financially, check out the official store or become a Patreon supporter with options starting as low as $1 or $2 a month. Anything you do to support the show really helps keep this whole ship going. I really appreciate it. I can't tell you how much I appreciate all of the donations and financial support that everyone has given me through the years. It's really been incredible, and I'm truly humbled. And as always, thank you so much for listening.